the banner of Indian Phytopathological Society. This is the third largest scientific society in the world. That's why we feel all the members are feeling proud of being members of this society. So in this webinar series, so we invited around 14 speakers lecture. So already we had one, and we are going to start now the second lecture. The speaker is this R. Salvador, and we expect very good lecture. He has chosen very nice topic. So here I would like to hand over this mic and this stage to Dr. Nalatambi. He is the General President of Indian Phytopathological Society, this Southern Zone. So here I request Dr. Nalatambi to brief about the biodata of our today's speaker.
Yeah, thank you, Dr. Indeed, it is a great place to invite Dr. Selvarajan, who is an eminent virologist, and of course, he is one of the leaders in my professional career as a senior. And sir, it is a great place for us. And to meet Dr. Selvarajan, he completed his undergraduate and postgraduate from Coimbatore, and completed his PhD from IIT. So right now he is working on banana viruses, and Dr. Selvarajan has got several awards and more than 10 awards, and to mention the few, and he was the and he got Class 1 Trust Award and FAO National Consultant Award, and he got best and fellowships from the societies. And Dr. Selvarajan was completed his degree program through the senior or junior research fellowship from the ICER. And currently his research area is plant virus diagnostics viruses, and he published several research papers and more than 20 papers and several publications.
Voice clear.
Echo.
Your voice is echoing.
Echoing, because I think Nalatambi used to only one person should be on in that room. If many are online, there will be a problem.
Yes. Or this is which
the room only one person should be online.
Switch off your mobiles.
Yeah, switch off your mobile. You should not come in mobile.
Yeah, mobile is switch off only. Yeah, now it's okay.
Yeah, now.
Yeah, okay now. Yeah.
Yeah. So could you hear what I spoke earlier, or should I repeat? Yeah, happy to introduce Dr. Selvarajan. He is an eminent virologist of this country, and presently he is working on banana viruses. And Dr. Selvarajan got several awards, and he completed all his degree program through ICER fellowship. And Dr. Selvarajan is specialized in diagnostics, particularly plant viruses, and therefore, since we are facing the virus disease in the human being as well as in the plants, and the society thought he will be the apt person on present day situation to give a wonderful talk on the diagnostics of plant viruses. Not only plant viruses, almost I think he will be able to cover other viruses also. And not taking much time, Dr. Selvarajan, we will welcome you. Kindly proceed your presentation. We'll after your presentation, we will come back to you again. Thank you.
Okay, thank you, Dr. Nalatambi Robin Gogoi, Secretary IPS. And first of all, I would like to congratulate the Secretary and the whole team of IPS sitting at Delhi, and the President also, and our South Zone President and the Councillor, for conducting this wonderful webinar series from IPS Society, under the IPS Society. And again, I am thankful for giving me opportunity to share my work with all of our IPS fraternity gathered today in this webinar. And I'll be talking the topic all of you know now. I think I will go for this sharing the screen. Yeah. Yes. Yeah. Okay, I am going to talk about the recent developments in plant pathogen detection, discovery, and diagnostics for deploying effective management against emerging diseases. Of course, I may touch one or two animal virus diagnosis also in this slides. So this, I think, is going to be useful for all our plant pathologists. And all you know about COVID. See, the virus which I put here is COVID-19, SARS-CoV-2 virus. Everyone know. I think all our pathologists, not only pathologists, everyone used to read about COVID. And COVID-19, as soon as it was discovered in December, immediately the pathogen was identified. It's a SARS-CoV-2, they named. Such a fast they made it. How they made it? Because of advanced molecular diagnostics, advanced molecular techniques to sequence the entire genome. So such a fast it could be possible. But whether I think all of you might be knowing about Spanish flu. 100 years back, a Spanish flu, which really threatened the whole population in the world. You know, 1/3 of the world population got infected with the Spanish flu in 1918. That time it was a World War I. Those people, it was not censored because they were not part of the World War team. So it was known to the world only because of Spanish people. Otherwise, it came from France to Spanish. That's what they say. That's what the name, it came as a influenza. Haemophilus influenza was the bacteria which actually, whenever this cold comes, you get a secondary infection. So they named actually.
Dr. Silvarajan, I am interrupting you.
No.
So you put on the slide mode.
Okay, please. Yeah. Okay. Thank you. Okay. All right. Okay. So that's what I'm telling. Spanish flu, it was not known. It came in 1918 and it left in 1919, killing 50 million people in a span of an year. And there was no vaccine. And forget about vaccine. It was not even identified. It's a virus. And that was the case. Now almost 1 million people people are going to die out of this COVID-19. And you know, because of this much developments, again, we are facing a problem because of this virus. And I would say virus identification is vital for the management process. And you know that virus, you know about Contagium vivum fluidum, the cause of mosaic disease of tobacco, that is by Martinus Beijering. In 1898, he proved that there is a live thing which can pass through the bacterial filter, that is Chamberlain filter, porcelain filter, which can retain all the bacteria, and it passes through, only the virus passes through that. That when you inoculate it, you saw it is multiplying, again it is causing this mosaic on the tobacco. Then they felt there is some organism which can multiply only in the living cells, and he named, and this is a Contagium vivum fluidum. So he said it's a fluid. But actually in 1933, when Wendell Meredith Stanley, a biochemist and virologist, he proved that this is a particulate in nature. In fact, he got Nobel Prize for that. So identification of a virus is very important. That's what now our the entire team in the world now searching for a vaccine, and we may come out with a vaccine very soon for this COVID. So here I'm going to cover the detection, discovery, and diagnostics has happened past 1 decade in plant virology as well as other pathogens also. So if you look at the detection methods, when 70 syndicator plants, when I joined in IRA, I used to just crush the tobacco leaves and Dr. Rangaraju and Dr. Mahmood Datta were teaching me how to inoculate and how to see locally and host, all those things we were doing. But now I don't know whether straight away our people go for PCR and look at the things happen. So these are the different methods what I have put it here, like indicator plan, transmission by vector, electron microscopy. Everyone cannot afford to have this. Of course, physically we can view. The visual diagnosis also comes under physical, the observation that also. So when you talk about diagnostics for viruses, it is only serological or nucleic acid. Serological because we are aiming for the code protein. Nucleic acid, we are aiming for the genome. There are many methods which I am going to talk in detail. And nowadays we get lot of WhatsApp pictures. Last 3 months I got more than 1,000 banana pictures, and a few of the pictures I have put it here. You see, this how can I identify these pictures? Immediately when I receive, our people, even our pathologists also want the remedy. What is the problem and the remedy immediately they want. Nothing else. What is that variety? Where from? What is the stage? Whether tissue culture or not? We don't have any other data, but they want the remedy. So is it possible to give by just seeing a plant and giving a remedy? It's very tough. But if you get a picture like this in WhatsApp, immediately we'll say it's a wilt disease. This is a wilt disease, and this is what we have to do. Just to go and download our app from NRC Banana about pest and disease management, you get everything about this. If you get picture like this, you immediately will say it's a bunchy top. Even it's a late infection of bunchy top, what you see in the flower, and we'll say banana streak virus. We may get pictures like this also. Immediately we'll identify this is a black mosaic virus. But 90% of the pictures what we get, we could not identify because we have not related the infection with this particular pathogen with the particular disease, or it is different from a nutrient deficiency and all. It's not documented that perfectly. So we need a very good diagnostics, means we need to have a good technologies. And I am going to talk about the detection methods which we have developed for banana pathogens. For developing immunodiagnosis, we cloned the code protein gene, we expressed it in a bacterial system, and we expressed it in a soluble form. That's more importantly a tough way to get a soluble form of the protein. Often we get insoluble this one, and we sonicate. Sometimes we send the insoluble fraction itself for raising antibodies. And we did for black mosaic virus, cucumber mosaic virus, and if you can see how it is coating, electron microscope, the antibodies which we produced could be able to decorate the black mosaic virus. And we developed ELISA-based diagnostics for black mosaic virus, cucumber mosaic virus, and we recently made a cocktail of antisera, and we made a single test because we want today a virus is there or not. It's not a matter which virus is there. So we made a cocktail of black mosaic and cucumber mosaic virus to do the indexing service for the tissue culture plants. And we also did for banana stick Mysore virus, wherein we took a viral associated protein because the code protein was not that clearly defined. There was a recent paper in IRI. But this one we did, and we got a very wonderful decoration of the virus particle, what you see here in ISEM. And we could able to do even immunocaptured PCR. In case of banana stick virus, the viral genome is integrated into the host genome. So unless, if you do straight away a PCR, you will get false positive. So you do an immunocaptured PCR, you get the right one, you get the episomal virus, not the endogenous virus, okay? So PCR-based diagnosis is a very common after serodiagnosis. PCR-based diagnosis is very common. In fact, lot of diagnostics developments have come now. Either serology or PCR. These two only adopted even in certification program. Most of the certification programs. And we did the black mosaic virus comparison, which is better and all those things. Of course, real-time PCR is best. What we do is COVID-19 real-time PCR only is being used actually. But can we use it for plant viruses? It's very tough. It's very costly affair. We cannot spend 4,500 rupees to test a banana plant whether black mosaic virus is there or not. So multiplex we did for banana bunchy top and banana stick Mysore virus using RT-PCR. You may surprise why reverse transcriptase PCR for BBTV. It's a DNA virus, banana stick Mysore virus DNA virus. So because to avoid the integrated sequences, we went for reverse transcription of both BBTV, and we were aiming the product, the transcripts of the viral genome. We aimed, and it was giving a wonderful result. And we multiplexed it. At a time, you can detect either of the one of the virus or both the viruses. And again, the same thing we repeated for all 4 viruses. We could achieve, and we could able to reduce the cost of the this test very low. But of course, this type of test should go into the certification programs wherein you can save the time and money, everything. So okay, this is the academic point of view. There are 6 components in one bunch of virus. So we could able to detect in a simultaneously all 6 components using a multiplex strategy. And yeah, I'm just jumping to change from virus to wilt. And Fusarium wilt, you know, it's one of the biggest problems, it's the biggest threat in India because it infected the Cavendish. That's what the biggest threat. Otherwise, Nanjangan Rasabali is lost. Our Rastali silk bananas are being lost. But this, when it is infected Cavendish banana, we are very serious because 60% of our Indian banana is around 60% of area is covered by this particular variety called Grand 9, and it's a very high yielder. It yields around 40 kg bunch. Average 30 kg bunch it yields. Almost if this particular variety vanished, we will be in a very big problem, and the entire world will be in a problem because this is the export variety. Autograss missile in 1960s ruled. This particular variety was introduced because it was resistant to race 1. But this became susceptible to race 4, that's called tropical race 4. For this diagnosis, if you very, very important, like viruses, here only 6th or 7th month you see the symptom. So unless you detect the fungus, this pathogen FOC from the soil or in a planting material like suckers, we are going to get this disease, and it's very tough to manage. And as you know, the clamdospores can live for 30 years in the soil. But can we detect such clamdospore which is living in the soil quickly? That is the question. And it's a challenge for all the plant pathologists that developing a wonderful diagnostics is will be very useful to manage this emerging disease. And you see here for tropical race for one of the group from Dita, this is he developed this technique from Netherlands. This technique detecting only the tropical race 4 exclusively. So it can distinguish between the race 1 and race 4 and all. But a recent paper says when they tested all 4 primers which is available in the publications, this is elongation factor 1 alpha, then specific TR4 also. When we tested, it is detecting the non-pathogenic Fusarium banana endophytes also. So non-specific positives, it's false positives were coming. So they tested all 4 primers, all published by different people, and they could not. They found only one, this W2987F, W2987R, could able to detect only the tropical race 4. So this is very challenging. A big challenge is ahead in developing this. And recently one paper came from Australian group, that is Caraval case, that is Aitken group. They targeting secreted in xylem genes for banana Fusarium wilt detection, and they made a wonderful thing. And I think all of you might be knowing that TR4 has been proposed a reclassified as a Fusarium vodarotism. This is 2019 they proposed, but I don't know the taxonomist has to accept. They came out with a wonderful this chart. You can see, we isolate the Fusarium, then do the DNA extraction, go for 6-1 PCR secreted in xylem, 1 gene. If it is positive, then go for restrictive digestion. Then if that is positive, it's a TR4. If it is negative, it can be either subtropical 4 or race 1. And for which again you go for 6-8 PCR. If it is positive, subtropical 4. So this is a wonderful way to distinguish tropical race 4, and race 1 and subtropical race 4. And a very recent paper, this is last year, September, it published this paper. And I think now one can go with secreted this one. And this, I was talking about multiplex. You see the citrus, our Abhijit Roy, he has developed a wonderful multiplex for citrus disease. At a time, 7 viruses of different virus genera can be detected. Okay, so this is multiplex is very useful, especially for citrus budwood introduction as well as citrus budwood certification. This is a wonderful technique which I think Dr. Dilip Ghosh and his team might be adopting these techniques. And again in grapevine, here also there are too many 80, 70 viruses are there. Here also some guys they have developed a wonderful for 9 viruses multiplex RT-PCR they have developed, and it works very well, and these things have to be utilized to save the cost and time. And for our Viswanathan group, they developed multiplex RT-PCR for 3 viruses infecting sugarcane. That's also a wonderful work from the SBI. And we developed a sensitive tag band based BBRMB. But you know, we can test, this is very sensitive compared to all PCR or all other methods. So the point is, we can detect even femtogram, 10 femtogram could be detected. You know, this can be, it's very costly, so we can apply for only germplasm screening. We cannot apply for routine indexing purpose. Okay, NAS-based diagnostics is very important, but since radioactive things we can give up, non-radioactive diagnostics can be used. And you know, very interesting is that polyprobe can be used. So when you use polyprobe, you can detect a multiple viruses. This polyprobe is, you clone all the small fragments into a single this one. Then you develop a probe, and that can be used. It's a very good technique, polyprobe, and it's very less number of people adopting this technique. And coming to the novel techniques, here only I am going to talk about novel techniques, which is distinct from the routine PCR method. So here I come lateral flow immunoassay detection system, and I think all of you know dipstick, you know immediately what you know, pregnancy test. So 25 years back, the pregnancy test, it came within 5 minutes, they will say it's a positive or negative. So that is human HCG, human chorion gonadotropin. That it's a glycoprotein, it's a hormone secretes when during the pregnancy, and that can come in the urine, and it can be tested, and immediately one can identify this dipstick it was used. And later in our in India, for BT cotton, Dr. Kranti, he developed a dipstick, whether it's a really BT cotton or not. So that was he developed, and really it was a wonderful tool to know whether it's a real BT cotton or not. So subsequently, there are a lot of work has been done on lateral flow. This is a wonderful on-site diagnostic kit. And you know, on-site diagnosis is very much needed for tree crops like citrus, apple, and grapes, and even banana. So wherever we need this type of crops, you can have a dipstick wherein you can do testing on the spot by the farmers themselves. A perennial tree and all, you have to safeguard from the virus infection. So let me say a little bit about this lateral flow, how it works. So you know, you have to have a wonderful anti-sera. If you have got a polyclonal or monoclonal anti-sera, very specific to your virus or any analyte, it will work very, very good. You have to isolate the IgG antibody or immunoglobulin of that polyclonal anti-sera. You can isolate and coat it here. That is test. And the antibody, what if it is raised in rabbit? You should have a goat anti-rabbit. The anti-rabbit IgG injected in the goat, what you get antibody, secondary antibody, you can put in the control. Okay. And this is very important. That is colloidal gold and the antibodies should be conjugated. The colloidal gold can be prepared in our laboratory itself. You just prepare this colloidal gold of different size, suitable size, and conjugate with that our antibody of the analyte which you are aiming. And if you are putting a crude sap here, this is absorbent pad is here, this is sample pad. So once you put this crude sap here, it moves laterally. That's what it's called lateral flow. When it moves, if there is a virus, it will bind with this, further it will move. If there is no virus, all things will be moving without binding in the test, and it will bind to this, because this antibody IgG can react with this goat anti-rabbit IgG. So it will form a line only in the control, but whereas test line it will not be there. If the virus is here, it will bind, and it will bind here. The leftover unbound colloidal gold antibody conjugate will further move, and it will show the control line. Very simple principle this is. you had to have a wonderful antibodies to develop this, and we developed for banana black mosaic virus, and we have also developed for banana cucumber mosaic virus, and we are validating with other crops infecting cucumber mosaic virus. And in fact, this was released by our ex-farmer agriculture minister. He released this kit, and furthermore, then we validated with many number of samples, and once we were happy and we published this paper. Recently, it came out in the Journal of Biological Methods. And similar technique, this is Xanthomonas wilt. Banana Xanthomonas wilt, a big problem. It's a quarantine problem for India. It should not come from Africa. So they developed this LFT for the Xanthomonas wilt also. So this type of kits can be developed not only for virus, but also for any pathogen, including Fusarium oxysporum also we can develop. So there are many LFIA kit available for plant virus detection. The one I think that because Mandal group, large cardamom Chirke virus, they developed a wonderful this one. And the same girl, Maheshwari, she developed for citrus virus when she went to Florida, I think so. This is like one has to develop these kits and it has to be commercialized for the use, because we Atmanirbhar Bharat, we have to develop and we have to deploy in ours. We should not import these kits from the abroad. So coming to rolling circle amplification, this is another wonderful technique, but you cannot use for that frequently for diagnosis. But this can be used for the discovery. This can be used for the discovery. Here, this P29 DNA polymerase, this has got strand displacement activity. And that's wonderful. And if you are adding just random hexamer, you need not to have any specific primer, you just add a random hexamer, then this polymerase, then you will get concatamers. You will get multiple copies of this virus if it is circular molecule only. So a lot of papers came in the Begoma viruses, Gemini viridae family, and we also applied for Nanoviridae and Badna virus. And you get such a good result here, and you can also develop infectious clone to prove whether it's infective or not. So rolling circle amplification is widely used for the discovery of viruses and not for a diagnostics purpose, not for detection purpose. So we did for banana bunchy top and banana stick Mysore virus. You can see multiplexing both. At a time you can detect both bunchy top as well as this one. Of course, once you amplify, you use a single cutter restriction enzyme, so you can find the molecule. So we recently discovered banana stick virus from hill banana that is caused by, it's a new virus, banana stick GM virus. Very interestingly, it has got only one ORF. Supposed to get 3 ORF. It has got only one ORF, and it shows the symptoms. But the thing is, this particular virus is not rampant. It comes rarely and it goes off. Probably because of this virus is mutated so much that it is not showing the symptoms. But it's not a big concern. But we identified this particular virus through RCA, then sequencing. And there are many reports, like our Mandal group. They did the cardamom butsi dwarf virus, Furki. This Furki virus, last 50 years, they said what is the causal agent was not known. As soon as this RCA came into picture, immediately our IRA group, that Mandal group, could able to detect and identify. And this is the next Babu virus in this group, cardamom butsi dwarf virus. And in apple, cocoa, a newly discovered virus, cocoa swell and shoot virus is well known. But cocoa red vein virus recently identified using the same RCA method. And in banana stick virus, there are 9 more additional viruses identified using this RCA by James et al. This is from QUT Australia, our James Dale group, but this James is different. Their group, they identified 9 viruses at a time, Diascoria also. I think Marquez can utilize this type of, I think it's already doing diagnosis using RCA. And coming to the loop-mediated isothermal amplification. This is a wonderful technique, and this is again a patented version, but it is being adopted in many places. Here you can finish very quickly. Here there are 6 primers you need: FIP, BAP primers. Then you have F2 and B2 primers, and you can also have a loop primer. loop primer. So it's highly specific. It will detect only the what you are aiming, not nothing else. So this is highly specific this one, but it is prone for contamination. If your laboratory is not clean, it can jump whatever the amplified product again and again. It will give a false positive. So you have to have a very safety laboratory rules to do this. Simply extract DNA, amplify and detect. Such a fast, and you can also do it visually. You can detect by fluorescence, and you can also detect by turbidity because it produces magnesium pyrophosphate that will give the turbidity. So based on the turbidity, you can say it is positive or negative. There are turbidometers available, real-time turbidometers are available. You can say real-time on this, you can take this small, because this works at 60 to 55 degrees centigrade, isothermal amplification. You can take to any place and in the field you can use it. And it's a wonderful technique. I don't know why it has not been adopted too much in the plant virus diagnostics. And we did LAMP for BBTV, black mosaic, banana stick, Mysore virus, cucumber. All 4 viruses we have done, and it's really sensitive. And you know, LAMP 1 picogram, 3.2 nanogram. So this has been done by other in China group also. They have done this work. And we also did for you need not to have a pure DNA for LAMP. Even if you do a simple DNA, we developed a simple solvent-free nucleic acid protocol, and we did a LAMP for Bunchita virus. You see, wonderfully we got, and in fact, we same thing we used for quantitative PCR also. So LAMP is a good technique which can be adapted for diagnostics. And you know, now LAMP with other techniques now they are putting to use. I will come and tell you about that LAMP connected with CRISPR-Cas related diagnostics. And you can see Fusarium oxysporum also they developed a wonderful LAMP to detect tropical race 4. This is by Zhang from Chinese group 2013 in classroom they published, and it specifically detects, and again it's from the soil. So you can detect a FOC tropical race 4 from soil using the LAMP technique. And of course, this can be made as a point of care diagnostics. And you see, there are many reports on use of LAMP, but none of them put in the use in the certification program. But definitely, I was not doing. And our Dr. Bhatt has developed cucumber mosaic virus, and again he himself developed for banana black mosaic virus. LAMP he has developed for his laboratory. There are many of our group have developed this technique, LAMP technique. And this is another important disease, you know, but of course not in India. Cassava brown streak virus. They did there are 2 viruses in, causing this cassava brown streak. One is Ugandan cassava brown streak virus and another cassava brown streak virus. They converted this into a dipstick. That's very interesting. So LAMP, you do it, then you convert into a lateral flow device. So a nucleic acid flow, lateral flow, nucleic acid assay. So this is a wonderful on-site diagnostic kit which can be done a similar method. Here they use dig probe or FITC to do this LAMP, to do this lateral flow immuno device. Okay, I will be telling about that. And another important technique is recombinase polymerase amplification. Very important technique, and widely used not only in plant virus but also in human and animal virus diagnostics. This RP is widely adopted, and in fact this was in 2006 this Pippenberg developed this RPA technology using proteins involved in cellular DNA synthesis, recombination repair. It's very interesting here. This is done at 37 to 42 degree centigrade isothermal amplification. You need not to do heat like double strand and all. You need not to do initial denaturation step and all. It's not needed, since this contains recombinase proteins, single stranded binding protein, and a polymerase. These 3 are there. This recombinase goes and binds with the primer and immediately recognizes the target, and it goes and binds. And when it binding to the primer, a D loop forms. That single stranded is bound by the single SS space, single stranded DNA binding protein. It will be, and it will be a continuous process of amplification using this recombinase, and very fast, 10 to 15 minutes, you will get a ample number of copies. And this is a wonderful technique, recombinase polymerase the amplification. Now it is commercialized by this Twist DX. It's the company, and they claim it's affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, delivered to the end users. So this formula, they use it for the human viral diagnosis and other anything. It's not only for virus diagnosis. It can be applied for many other pathogens. And in fact, recently, this RPA, they have applied for the Fusarium oxysporum cubens tropical race 4. And one of the foreign company, I think, Artia, has come out with a kit, on-site diagnostics kit for Fusarium oxysporum cubens, that is Fusarium built. So that in June only they have released this kit, that is RPA based kit for early detection of FOC. And we developed this RPA for BPTV and cucumber mosaic, it's a DNA virus, and another is CMV. So we did both. We did reverse transcription, then RPA. So reverse transcription, then followed by RPA. We did, and we could able to detect simultaneously both CMV as well as BPTV together. And this why we targeted was for grandin banana. These two viruses are very omnipresent. So we thought these two viruses could be detected, and we are going to convert this into a lateral flow device, RPA, and followed by lateral flow device using CRISPR. We are attempting. We will be coming soon with the lateral flow for BPTV and CMV with the nucleic acid. This one. So all you know about this CRISPR, all we talk about CRISPR, genome editing, CRISPR. I think not only biotechnologists but also pathologists, everyone talks about this CRISPR. CRISPR is basically a clustered, regularly interspersed short parenzymic repeats, and you know it was in a bacteria. It's an adaptive immune system developed in a bacteria, and to control against the invading viruses in the bacteria. And you know you can see this repeated small pieces of viral genome followed by host genome will be there. And here very interesting thing is it has got a rec, that is recognition lobe, and nucleic nucleus lobe. There are two lobes. Cas13a it produces, and this is a pre-CRNA, CRISPR RNA. Once this CRISPR RNA goes and binds in the recognition site, then it makes some conformational changes. So once it makes the conformational changes, this nucleus will look for a target RNA. Based on the guide RNA, this target RNA will be there. So once target RNA it recognizes, nucleus will be active. So nucleus will be cutting this target RNA. Okay, very specifically it will cut the target RNA. That will be a PAN sequence. A protospace or adjacent motif will be there. Then there will be a 20 bases. It will cut exactly where you want. But while cutting, it has got a collateral nucleus activity. That's very important. It's a collateral, non-specific nucleus activity also will be there when it is recognizing the target. That you know that it will cut the any single-stranded RNA molecule in case of CRISPR-Cas13a. It will cut the RNA molecule if it is single-stranded. In the nearby area, if it is there, it will be cutting. So utilizing this, they have developed a wonderful diagnostic. This one you can see here, double-stranded DNA. Then you see the target RNA, target double-stranded DNA is here, and this is small, just 10 base pair reporter. 10 base pair reporter is there here, wherein a quencher will be there, fluorophore will be there. A quencher, fluorophore, single-stranded DNA is there. Of course, this is LB-Cas12a. In LB-Cas12a, single-stranded DNA is a reporter, whereas LB-Cas13a, single-stranded RNA is the reporter. Okay, so once this target site is recognized, immediately it will start non-specific cleaving of this reporter. Once it is cleaved, you see the fluorescence. If you have got a fluorimeter in your hand, immediately you can say it is positive or negative. But unless there is a recognition, this will not be cut. So if there is no target, there is nothing. So you can see here African swine flu virus, they have detected. You see what a fluorescence here, but it is not detected with the other related viruses. This is very, very recent. Just a last month this paper came. So I want to share how CRISPR-Cas is being used for diagnostics, and they converted this into a lateral flow. So this can be converted into a lateral flow. So just whenever we put in the sample, you put this product into the sample pad, then you can do it. Here you had to have big oxygen, biotin, streptavidin, anti-mouse antibody, then again gold nanoparticle. So gold nanoparticle and CRISPR-Cas12a product. If you put, you will get this. So this African swine flu virus, they adopted, but for plant virus yet to come. I think we are all working in this line. Either you can, yeah. But this is our Arun there. I think you might be knowing one of our virologists who is now working on white spot syndrome shrimp virus. And he developed, recently it came in the scientific reports, he developed, it's a DNA virus actually, this white spot syndrome virus. They converted, they did an RPA and they did a T7 transcription. They made RNA. Then they used Cas13. Had they used Cas12a, this transcription would have not needed. T7 transcription would have not been needed. So had they used Cas12a. So they used this guide RNA Cas13. Then they reported with RNA. This reporter is single-stranded RNA. So they converted this. Again, this Dr. Arundar group, they converted into a dipstick and they made a on-site detection of white spot syndrome. This also appeared very recently. And again, very, very recently, just 10-20 days back, this appeared. This is detector. Here, O is missing. It is not O is missing, it is their trademark. They detected this, our SARS-CoV-2, very fast they made this, and they aimed envelope protein, and they did this nucleoprotein. Here, they had to initially do a LAMP, loop-mediated isothermal amplification aiming E envelope gene and nucleoprotein gene, either this or that. You do LAMP. Immediately after doing LAMP, this nasopharyngeal swab you can take, extract the viral RNA, do the LAMP. Then once you do the LAMP, you add this CRISPR-Cas12. Once you add, if the target is there, it will cleave the single-stranded DNA probe. So this, once it is cleaving, it will fluoresce. If you have got a fluorimeter, immediately you say it's positive or negative. If you don't have, you can also convert this into a dipstick, like lateral flow nucleic acid test. So CRISPR-Cas has been widely adopted to do the point of care or on-site diagnostics, and this can be applied for many of the tree crop viruses. We can apply now. And you come to the discovery of the viruses. You see a wonderful picture I am showing here. This is a gymnema. You know, it's a gymnema sylvestris plant. We saw this. Immediately I uploaded in the WhatsApp group of virology group. They said our Dr. Parak and Dr. Paul Kuranasab, both they said this is CMV. Immediately I trusted and did an ELISA. It was not CMV. Then I was thinking what to do and all. Then I somehow, my intuition felt that this may be this groundnut bud necrosis or tomato spotted virus. And when that time I contacted Dr. Vichi Malathi and Dr. Vichi Malathi and his team, and we did this using the tomato spotted virus degenerate primer. Then we got positive. So we inoculated in the tobacco, also it became positive. So this was collected from a backyard of a single house. I used to go in the morning walk and I collected this leaf samples and I did this. So tomato spotted wilt, this is almost 3 years back, 3-4 years back. Tomato spotted wilt virus in gymnema has been identified, and I don't know where from it came. Definitely it is not from abroad. It is existing in India. Just by while going walk, I could able to identify a TSWB infecting gymnema. So similar thing I am going to tell you. So another discovery, if you talk about, sorry, one more, yeah, it's missing. This is a high throughput sequence equipments. You can do a lot of discovery, new viruses. either this Illumina, iron torrent, they do sequence by synthesis or single molecule synthesis by PacBio or Oxford Nanopore. So these can be exploited for discovering a new virus. And you see here, very recent paper appeared in China paper, from Chinese group, they published this paper. It was very interesting. So very wonderfully they have worked with this. If you show these pictures, immediately our soil scientists will say, this is due to some micronutrient deficiency, iron deficiency and all. And if you show to some other people, this yellowing something, nothing, no virus, nothing. But if 26 samples they collected, they did high-throughput sequencing using metagenome sequencing using Illumina platform, and they came out with 7 viruses. So they got 100 GB data. Finally, 2 GB data was related to virus, and the 2 GB data they took it and they identified blueberry necrotic ring blotch virus. Another is American plum line pattern virus. They renamed as tea plant necrotic ring blotch virus, tea plant line pattern virus. These 2 they cloned, sequenced, they confirmed by ordinary other sequencing method, they inoculated, and they got, so one is Hilar virus, the second one, line pattern virus is Hilar virus group, the another I'm not remembering. There were 36 species of viruses in that high throughput. Out of which these 2 they proved everything and they got whole genome sequence. They got, so discovery could happen within a month or 2. You can detect what is the causal agent is present. And you see, they have applied, actually in plant virology, 2009 onwards, they applied this technique. Using high throughput sequencing, can we detect and diagnose or discover virus? Yes, it was possible. The Adams 2009, he used Liatris picata symptomatic. They found a new virus called Cape feather mild mosaic virus. It's related to cucumber mosaic virus, the bromovirus. And similarly, there are many viruses they discovered, around 15 to 20 viruses using the high throughput genome sequencing method. And you know, there are a lot of, I don't go in depth on the techniques because you have to use either small RNA you can target, you can go for double strand RNA or anything. Total RNA you can use. If you are using total RNA, you have to eliminate the ribosome RNA and you have to enrich the polyadenylated RNA. Then you will, the chance of getting the virus is more. And there are a lot of pipelines are available. So you get a huge data. How do you handle? So Linux-based data pipelines are there. You just give an input and it will stepwise, it will be working out. Virus hunter, virus seeker, virus finder, and there are many truffle. And again, web-based GUI, the graphic users interface. Okay, this virus find, virus detect, VST toolkit. This can be adopted, and there are website links there. One can use and we can analyze. Bioinformatics is very, very important in next generation sequencing. Unless our pathologist, probably this, I think, training one has to give in this line to do more number of people to work on this line. So, using transcriptome analysis, we did for a banana called Puvan, Mysore, and we found tobacco mosaic virus in it. Tobacco mosaic virus. Then 20 years back, Dr. Singh has published a book in which he has mentioned about tobacco mosaic virus in banana. But it's not there very rampantly everywhere. But we could be able to see in the high throughput sequencing. Then again, banana stick Mysore virus, we got very, you see, read that we got very huge number of reads. And we got the entire genome. And we got banana bunchita, all 6 components of banana bunchita virus we got. And we could also see what is the ratio of each component in the sequence we could see in the HTS. This was done in Illumina platform. Okay. Then we also did for banana brag mosaic virus collected from Assam. This also we did, and we found out of these many high quality reads, we could able to get the entire virus with a very good sequence data we got, and we have communicated this paper, banana brag mosaic virus using NGS platform. And the recent, we bought this MinION Nanopore sequence, very wonderful. This can run in a laptop, this particular instrument, you can run in the laptop. There is no need of external this one. This, you know, what is this? This is E. coli channel protein. bound in a electrically resistant base, and it's very wonderful nanopore. This one, Oxford nanopore instrument, could be used widely for diagnostics, for point of care or on-site detection. Here you have to isolate DNA, then do fragmentation like for other also. It can be enzyme digestion, then end repair, qubit QC you have to see, quality control, then barcode. You do barcoding, barcode ligation. If you have got multiple samples, you do barcode ligation, do the adapter, then you load it, flow cell priming, you do it, library you can load after priming the flow cell, then start sequencing. Once you get a lot of data, you need to go for base calling, then demultiplexing, then QC trimming, mapping, then consensus. So we did this particular nanopore sequencer for banana. 3 samples we took, and we could see, you see how we could totally map the entire 6 components of the virus, and 99.9% accuracy we got, because the number of reads we got very high. And another very important thing is, when you use genomic genomic DNA, it's very less reads we got. When we did a RCA, enrichment through rolling circle amplification, we got very huge number of reads. And using genomic DNA, we could not do characterization part. But when we did RCA product, we could characterize the whole genome, whole sequence we got, and it was very, very almost equivalent to Illumina data actually. And we did again, this is one of the CRP project, vaccine and diagnostics, where Dr. Krishna Reddy is our coordinator, and we combinedly joined with the sugarcane group and our Dr. Makesh from CTCRA, their group and our group. We joined and we used the multiple DNA viruses, and we got Sri Lankan, what Dr. Makesh Kumar gave, a single sample contained Sri Lankan cassava mosaic virus as well as Indian cassava mosaic virus, and banana streak virus, Begomo, Bunchy top, Jatropa leaf crumple virus, cotton leaf curl, Multan virus, I don't know where from it came, bendy yellow vein virus. So of course we had to do further study in this. And very interestingly, this nanopore sequencing we applied for RNA virus also. Here we collected from Dr. Krishna Reddy lab, we got chilies, tomato, tuberose, papaya, and sugarcane from SBA, black gram from Tamil Nadu Agricultural University. They have got a black gram leaf crinkle virus. It's one of the big problem. Then sweet potato, diascoria, MF, banana, all these things put together we got this one. And in this, we got tomato spotted wilt arthovirus from tomato sample. And this is very interesting. This is the first report that TSWV could be detected from the tomato. And when we received sample from IAHR, we were not knowing this will be coming or not, but this came. And cucumber mosaic virus, bragg mosaic virus, papaya ring spot, all those viruses we could able to detect. But of course, the method which we followed is only for detection, not for characterization. The method for characterization, getting a full length genome and all, it's a different. That also we have done and will be doing. And again, this nanopore sequencer can be adapted. We did a spiking with Fusarium oxysporum with the banana virus, and we could able to detect. So it is possible to collect a root sample infected with the virus as well as infected with the Fusarium and make it DNA, and you can detect on the spot if it is there. So nanopore can be applied for multiple pathogen detection also. And you can see the nanopore has been applied for in yam, tomato, yam. This 2018 paper, I think that Makesh Kumar also one of the author of nanopore based sequencing. Tomatoes and wheat, potato, coffee, around 4 or 5 papers, just last 2 years it came. And very interesting nanopore sequencing technology we can adapt for on-site detection as well as characterization. So actually at NRC Banana, I want to just take a slides that we developed a lot of virus diagnostic kit at our institute. ELISA, DIBA, RT-PCR, PCR, NAS, and we have given list of we have got only 4 viruses. Of course, banana stick mild mosaic virus is there. We have reported for the first time, but that's not an important virus. See, we have developed Recombinase, RPA, lateral flow, loop-mediated, qPCR, multiplex, NAS, all we validated with a huge number of samples. And one has to adapt this technique for the certification. And last 2003 onwards, we are doing 2003 onwards, we are testing for mother plants collected from tissue culture companies. And you can see down this graph carefully. You can see this red is positive. See, they send the sample without no symptom, nothing. It's all pure, means very healthy plants. And I would like to share one important thing. Okay, I think next slide I will be telling in that. So here we received a lot of samples from the Jam Blossom from Asia, tropical fruit centers from Arabavi, Coimbatore, Gadevi, Jorhat, Jalgaon, and we tested 1,650 Jam Blossom, and we found a lot many samples. Accessions were positive for banana black mosaic virus. In fact, we tested by dipstick also, and it was positive. So we were doing this testing for long time, since 2003. But the certifications started where we were giving only last 3-4 years. Around 300 million tissue culture plants we certified from 93 tissue culture producing company. But before that, we were testing, but we were not issuing any certificate. That if you count, it is equivalent to 622 million tissue culture plants. Totally around 1 billion tissue culture plants, banana plants we have certified, I would say. So that's one of the biggest achievement of our center. And we are even now doing testing, not as an accreditation lab, but after we get a lot of samples and we test. And another important thing is banana bunchita virus in the hill banana. That's the virupoxy, the biggest problem. Whenever you go to lower Palling Hills, 60% banana bunchita we can see in 1970s. Now, whenever we go, at least from 20 to 30% every orchard you will get bunchita. So we started collecting. Once we collected 200 mother plants, all are symptom free, nothing was there. When we tested by PCR, and we found 120 positive, only 80 were negative. So we gave to that 120 plant, we removed only 80 negative, we gave to one tissue culture company, started multiplying, and that company, out of 80, there was one false, I would say it's a false negative. That was actually positive, one more sample. So the company called me, why this all bunchita? So around 800 plants are bunchita. So out of 80 mother plants, they used one escape. Because as I said, there are 200 mother plants. When they took, 120 were positive. So they had to eliminate such a big number, and they established the virus-free plants, and they will keep on giving. And there are many companies now, they are doing around 6 lakh bunchita free virupoxy plants have been supplied by Tamil Nadu. Of course, the involvement of Tamil Nadu, government of Tamil Nadu, that is Hill Banana Rejuvenation Scheme, they deployed, they give free of cost. This particular virus-free plants are being given free of cost with the help of this Tamil Nadu Banana Growers Federation, Hill Banana Growers Federation. They are also working a lot. They got Plant Genome Savior Award for protecting this virus, this particular variety, Hill Banana Virupoxy, from bunchita. Otherwise, this would have been extinct, this particular variety, because of bunchita. So, bunchita is only a virus which can kill the plant, not like black mosaic or streak virus. So, this is one of our achievements, that we could able to rejuvenate the GI tagged Hill Banana Virupoxy. And again, for Puvan is a big problem, and we using the diagnostic kit, we developed a virus-free, episomal virus-free banana streak Mysore virus, and all have been tested in the 5 ACRP fruit centers, and it is outperforming, and none of the plants showed any episomal virus. That's very interesting, and yield also very higher than the sucker grown plants. So this is one of our achievements. We got virus-free plants. In fact, we got it from 15 years old permanent field trial, where from we were testing continuously for 3 years. We picked up virus-free plants and multiplied, and we have got this virus-free stock with us. And I think I have completed in time. This is for now. I think I will stop sharing this. Yeah. Thank you very much.
Okay, thank you, Dr. Selvarajan.
Thank you.
So, according to me, this is called not a single stone unturned. So we have received so many higher appreciation on your lecture. You have covered each and every corner of your this own subject. Even you have reached the fungal pathology also to some extent. So really, it is really appreciable, and you will be happy to know that more than 375 participants already joined.
Yeah.
And we don't know from YouTube how many people were watching you.
Yeah.
So my dear viewers, audience, and IPS members and others, so I would like to hand over this platform to our South Zonal Indian Phytopathological Society, this councillor, Dr. C. Uma Maheshwari, to place vote of thanks for me.
I would like to tell one
Yes,
thing.
yes.
I think,
Sorry.
yeah, there will be a lot of questions would have come in the chat.
Sorry. Here, before going to this vote of thanks session, we have received 8 or 9 questions, but here I think at least 3 or 4 questions we can have discussion.
Yeah, 1 or 2 you can. Yeah, that's
Yes,
what I.
yes.
Yeah. 1 or 2 questions.
Let me check, and I'll name one by one. At least 2 or 3 maximum will be accommodating, and rest I request all the participants who are having some questions to contact Dr. Silvarajan directly through his email address. Okay, if you don't mind, please.
Yeah,
So
yeah.
now first, in the first question, let me choose. Here, Sipra Sharma. Are you hearing Sipra Sharma? So, Sipra Sharma is asking why there is more mutation rate in viruses than other microorganisms. I repeat, why there is more mutation rate in viruses?
Yeah. Wonderful question. And you know, more mutation happens in RNA viruses than the because it's error prone. It's error prone. It's well known. A lot of documents are there. So virus is prone for mutation. And you know, during the mutation, it is only the one which will be surviving. Is lethal mutants will be it will be escaped from the population, and the population. The diversity of the virus, a lot has been worked on it, and viruses are highly prone, especially RNA viruses are highly prone for mutation than the DNA virus, since it's a error prone mechanism in the replication.
So second question. So this question is from Dr. L. Dayo from Nagaland, Meji Thomas Sassard. Okay, he's asking, Naga king chili is a potential commercial crop. Farmers are facing a big problem with viral diseases in king chili. What effective methods our farmers can adopt to avoid the viral diseases, which is also a common occurrence on other chili?
Yeah. King chili, yes, I know. I have also tasted that. I think in Assam Agriculture University, people are working on this viruses affecting king chili. But of course, as you said, we have to give virus-free. There are 1 or 2 seed-borne viruses also there in chilies. But I don't know in king chili what all the viruses are. Of course, I know that cucumber mosaic virus infects king chili. That I know very well. So it is possible to have the seed material free of viruses and other things. And otherwise, if it is in the field level managing, it is possible to take care the aphids and all which is transmitting the virus. So going for an insecticide spray, if the inoculum is very high, it's better to go for imida like insecticides to control the vectors so that the spread can be avoided. Otherwise, having a virus-free field is very, very important. That could be achieved by diagnostics, effective diagnostics, deploying effective diagnostics. Yeah.
Now I'm allowing Namita PM. So Namita So you can put your question directly to the speaker. Namita PM. Are you hearing Namita? Are you not connected properly? Next.
I think do we need to specific primers for each virus for using LAMP for detection? Of course.
Yes.
Yeah, that's very much needed. See, if you want to do multiplex in LAMP, it's very tough actually. If you do RPA, if you are doing RPA, you can do multiplexing easily. But if you are doing LAMP, multiplexing is little tougher, and you have to have specific primers for each virus when you do for multiplexing. Yeah. And another question, should I anonymous attendees should we talk about that? Could you please explain Western blotting with the polyclonal antibodies produced by synthetic peptides for virus detection? How sensitive it is the question. Okay, definitely it will be very sensitive. Synthetic polypeptides means expressed peptides. If you raise antibodies for the expressed protein, and if you do Western blotting, definitely it will be very highly sensitive. It can able to identify, but Western blot cannot be applied for any diagnosis purpose. Then coming to the Rasul Rather, is there any possibility to detect some important fungal disease like apple scab, which are required to be detected in plant system during their incubation period before symptom development? Detection and identification after symptom development is just useless. Yeah. See, like choletrotrichum also, like apple scab, choletrotrichum also latent in banana. So, can we detect little early this choletrotrichum latent infections because it affects the export in banana? Anthraconosis affects in export also. But of course, there are no methods available to detect. But otherwise, it is a candidate that one can work on this apple scab as well as choletrotrichum to develop a good kit to detect during the latent period. Thank you.
So here we are happy that our president, Dr. P.K. Chakraborty, has joined very recently. So he's very much eager to hear Silvarajan, but he was late. Anyway, I invite, so he's having some questions. So here is Dr. P.K. Chakraborty.
Yeah.
Congratulations, Dr. Selvaraj.
Thank you, sir. Thank you very much. Thank you.
In fact, Dr. Gogoi knows we are in another thing. We are very much grossly engaged in that, but it is nice to see. In fact, from days together, I have been making up my mind to listen to you, and it was one of my pet topic. And this morning, just now, with our chairman ASRB, I was discussing on this diagnosis, and it's important. And you have really touched a very important topic. And being International Year of Plant Health, that was really good. And the first topic I just read about it, Dr. Velahan, he presented about detection of aflatoxin. That is also equally important because every year when we go to represent India in Codex Committee on contaminants in food, aflatoxin detection, aflatoxin is a big problem. However, I missed that, but I could listen to your last part. I have only simply one thing. I know the future is in good hands regarding diagnostics. One thing, a kind of one clarification, or a comment that in India, diagnosis has become a fancy word, you know. And its use, its real use in epidemiology, has not gone to the real site where to detect them at the source, so that plant protection measures can be initiated from where it has come. But one more my suggestion would be: has there any work on e-noses? E-noses have been widely being used in quarantine purposes in Australia. So now you are such a wonderful scientist, and with your team in NRCPP and other places. So you may lay some hand and start beginning, because that's a quarantine issue.
Yes,
And
sir.
whether tropical race 4, although your area is mainly on virus
Yeah,
disease,
yeah,
I believe.
yeah, yeah.
But then even then, there's a host pathogen interaction resulting into certain release of volatile organic compounds,
Yeah.
and based on that, certain countries, there are hundreds of e-nose modules have been developed, and many countries are already developing, using them in their quarantine, like New Zealand, Australia, and some other.
Yes.
So, how far are we from that kind of studies to be initiated, and why not you can start initiating some studies on in that area?
Yes, sir. Yes, sir. Yeah, sir. Thank you. Thank you for coming in live. Yeah, it's a really wonderful question which you have asked. But he knows, yes, tropical race 4 is emits some order. It emits some order. Definitely, it's a good candidate to go for differentiating Fisherium moderotism versus the rest of the races through ENOS 1. Number 2, this very much, and of course, we have not put any effort to do this particular job. Definitely, we will put in. And another important thing I would like to tell you is this Aphis gossipy is not a host. It will not infect banana actually. But whenever banana bunchita virus is there, infected plant, this Aphis gossipy can come and colonize, and it can reproduce very fast. So a non-host it becomes. So its behavior of the vector is being changed because of infection of banana bunchita virus. So we did some GC-MS work, the volatiles, whether it is differing and all. We got some basic data now. By further analyzing, we can come out with a ENOS for infection, and this will be useful for epidemiological work as well as field level diagnostics work also, especially for wilt point of view. We can do field level diagnostics and differentiating tropical race 4 with the rest of the this one. And a very good suggestion, sir. Definitely, I will put up to my director, and we will try to start this particular type of research in our
Yeah.
institute. Yeah.
Thank you, Dr. Selvaraj. And
Thank you, sir.
this will definitely attract funding. No, you can
Yeah,
attract
yeah.
funding from
Yes,
DST,
sir. Definitely.
not only from ICR but also from DST, because
Yeah.
the volatiles, you saw that there is these two host pathogen interaction, banana bunchy top and with banana, has
Yeah.
resulted into develop on the release of some kind of volatile organic compound, which if can be characterized,
Yeah.
then it can show that there will be predisposition of aphids, and
Yeah.
finally there can be a timely measure can be initiated for the management of aphid as well, in general.
Yeah.
So thank you so much, and
Thank you,
for
sir.
a nice presentation.
Thank you, sir.
Unfortunately, I missed most of it.
Yeah.
But then the way I see the congratulations pouring in, and
Okay.
and the people's remarks pouring in, you are wonderful. You had given a very good talk, and thanks for the efforts of our IPS Secretariat for taking up this unique job of webinar. Thanks, Dr. Robin, Dr. Kollan, and Dr. Gujar, and Dr. Rajkumar, and Mr. Rajkumar is there, Raju. And thank you all, and thanks all the participants. That's from my side. Thank you so much.
Thank you, sir. So here we will be having only one question. Last questions from Dr. Krishna Reddy.
Oh.
Dr. Krishna Reddy is here. Last questions. So I get pardon from other questioner. Last question we want to have from Dr. Krishna Reddy.
Yeah.
Oh.
Selva.
Yes, yes, Dr. Krishna Reddy. Good morning. Good morning.
Good morning. Nice talk and nicely comprehended. As Dr. Chakravarti was pointing out,
Yeah.
the real usage of diagnostics in Indian conditions, you should highlight that at least in the diagnostic platform,
Yeah.
diagnostics were used in quarantine, and the export of horticultural crops in India is contributed through diagnostics only. The entire quarantine export in India is only through testing by our service.
Yeah, of course. Yeah, Dr. Krishna Reddy does lot of testing for ornamental and banana, and there are many crops. He does this testing
And-
for. That's very
And
wonderful
my
service he is doing.
question is, technical question is, the Nanopore or NGS sequencing system,
Yeah.
when you do sequencing, you get lot of sequences.
Yeah.
What is the limit you take it as a positive for presence of virus diagnosis? Percentage sequence reads in a total sequence reads.
Yeah, one thing, sir. One is, as you said, the reads. Number is the read length. Because in single molecule sequencing, especially nanopore sequencing, we get a read length of entire genome also we get in a single read length. So both read length, number of reads, both important. So for a quick what's in my pot, if you look at the software, there's a called what's in my pot that is there. It will give you immediately the number of reads, and if you map, you will know the sequence coverage, based on which we can taxonomically we can look for that particular sequence, like replicase genome, replicase gene in case of the cheetah, like virus. If you want, you can go only for core protein. So that particular thing, if you take, you can say if positive or negative. So number of reads, even 10 will say positive.
Thank you.
Thank you, sir. Thank you.
So, thank you. Let's proceed to our next agenda item of this talk. So
this will be available on YouTube also.
This will be available on the YouTube also.
Yeah.
So one can access from there.
Yeah.
So if some questions are still left, so
Yeah, I can answer these questions, and I can pass it to IPS.
Yes.
Okay, is it okay? No, there are questions I could see. Questions, there are around 21 or unanswered questions are there.
Right.
So this can I download these questions? I don't know.
No,
Or
yes,
you can pass it. I think IPS
Yeah,
you can
we'll
download and
Yeah,
give it to me. I will reply back.
Yeah, we'll provide you if you cannot download. So we'll
Yeah.
provide you.
Yeah, kindly provide. I will answer for every questions.
Okay.
And again, I thank see joint secretary sitting there. He was not there in the meeting.
Yes,
So thank you. Thank you.
So here, this is the last part of this talk. So I request our Zonal Councillor C. Uma Maheshwari, Dr. C. Uma Maheshwari, to place the formal vote of thanks.
Dr. Gogoi.
Sir?
Hello.
Because what we had decided that a set of recommendations should emanate from all these presentations.
Okay.
So maybe finally some practical points should come. One or two solid this recommendation should come. Either in the form of diagnostics, which should be used universally at what point and all such kind of thing, or some kind of policy parameter, policy thing, which can go as a policy thing. So that at the end of all the presentations, we will we have to compile them and publish a kind of booklet of recommendations to be passed through ICR to DAC for implementation and other advancement wherever we might have to look where does it go, whether they are for policy issues, whether they are for some other issues, whether they are for management issues. So all those kind of things needs to be done. So some 2-3 recommendations coming out from each of these presentations, maybe from the speaker itself or those who are rapporteurs who are there, they can draw and try to give it a look that it goes to either policy or some kind of reporting of advancement in the country. So that should go, and we will have a very beautiful publication which we can release at the time of maybe immediately after the annual conference or before, can be placed in the annual conference itself. So that further, so that we may decide. Okay, so kindly note down few recommendations which are coming out from each of them.
Thank you, sir. So here definitely we will be compiling those recommendations, and also in consultation with each of the speaker, we will finalize the recommendations.
Yeah.
That it should not be from one sided; it should be from both the sided, speaker as well as IPS side.
Yeah.
And that way will be definitely compiling some recommendations so that that can be executed in future for the benefit of our crop protection scientists and also crop protection.
And policy, as such for policy
Policies
related
also.
issues.
Sir, yeah, really, yeah. So thank you once again, sir. So I request Dr. C. Uma Maheshwari, this zonal counselor, to put this formal vote of thanks.
Hello. Good morning to all.
Yeah, கேக்குது.
Hello. Hello. Good
Yes,
morning to all. So I think successfully we completed 2 lectures. So on behalf of IPS President Dr. P.K. Chakravarti sir, and also on behalf of EC members, and especially from South Zone members, we thank 2 speakers, first Dr. Selvarajan sir and also Dr. Velalagan sir for their very good lectures. And when we approach them, they've given their consent immediately. And so we all got this opportunity to listen their lectures. And it is very informative. And I thank all the organizers, Dr. Gogai and Dr. Kalyan Mandal and others, Raju and Malkan, to organize this webinar seminar so successfully, even under this COVID situation. And they are instrumental for this successful event. So I thank all the EC members and all other organizers and our President IPS also, and all other panelists and the other participants for this session. Thank you. Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye. Thank you.